Today topic of discussion is carbohydrate metabolism. Carbohydrate are main part of our food. It is presented in the form of polysaccharide, disaccharide and monosaccharide. After digest it will converted into simple sugar like glucose, galactose and fructose. Glucose enter four type of metabolic activity: glycolysis, glycogenesis, glycogenolysis, glyconeogenesis. So already glycolysis over. This one is the second process, glycogenesis. Now we will discuss about what is glycogenesis. Glycogenesis means synthesis of glycogen, the conversion of excess glucose, to converted into glycogens. Excess of glucose is converted into glycogen by liver. This process takes place in liver. So conversion of glucose to glycogen, the process name is called as glycogenesis. It takes place in liver. Store extra glucose as glycogen in liver and muscles. Many steps. First steps are glucose is converted into glucose 6-phosphate. Here phosphorylation will take place. So adenosine triphosphate is converted into adenosine diphosphate. One more inorganic phosphate will attach to the 6th position of glucose molecule, carbon atom. So glucose 6-phosphate. This reaction catalyzed by two type of enzyme. Glucose is presented in the muscle means hexokinase. Glucose is presented in liver means glucokinase. So two different type of enzyme catalyzed this reaction. Glucose is converted into glucose 6-phosphate. Then with the help of phosphoglucomutase enzyme, glucose 6-phosphate is converted into glucose 1-phosphate. Here what is the role of mutase? It will change the position. Carbon number here 6th position, here it will change to the 1st position. Isomerization takes place. 6th position transfer to 1st position. Third one is uridine diphosphate glucose is synthesized from glucose 1-phosphate and uridine triphosphate by uridine diphosphate glucose pyrophosphorylase enzyme. With the help of uridine diphosphate glucose pyrophosphorylase enzyme, glucose 1-phosphate is converted into uridine diphosphate glucose. This one is the glucose molecule 2-phosphate. So diphosphate, this one is uridine. Now initiate this process required primer. Pre-exciting small fragment of glycogen is act as a primer. Glycogenin can accept glucose from UTP glucose molecule. This glycogenin accepted this UTP glucose molecule. The enzyme glycogen initiator synthase. Glycogen initiator synthase transfer the first molecule of glucose to glycogenin. Glycogenin synthase is responsible for the formation of 1,4-glycosidic linkage. This one make linear molecules with 1,4-glycosidic linkage. Formation of branches is brought by the action of branching enzyme 1,6 linkage. So 1,4 linkage we get that linear. With the help of 1,6 we will get that branching. So finally we will get this glycogen. Glycogen is further elongated and branched by the enzyme glycogen synthase and branching enzyme. We will get glycogen. So glucose is converted into glucose 6-phosphate, then glucose 1-phosphate, uridine diphosphate glucose. Then we need then base. That primer, it will accept the glycogen accept uridine diphosphate glucose. Then with the help of glycogen initiator synthase, it will accept. Then it form 1,4 linkage. Then with the help of branching enzyme, it make 1,6 linkage. Finally, we will get linear and branching elongated structure of glycogen. Notes format. Thanks for watching.